Hello everyone. Today we're going to be talking about the final section on history here of DNA, the discovery of the structure of DNA. I'll also link up a video or a documentary that will cover some of this idea as well, especially Rosalind Franklin, who is should be and it is now a celebrated a female scientist from the 1950s, and her work was highly underappreciated at the time, and only now is being looked at for the amazing accomplishments she made, not only in DNA but also in the study of viruses. So now that chemistry was understood about DNA, there is some wonder about how could these chemicals, these nucleic acids that Chargaff and the rest had so carefully studied, how could these things be actually storing information? And we have to also remind ourselves that we're still in the 1950s. Computer science is a developing field, so the idea of learning how even the method of storing information is actually it's still on the growing. It's still growing. This concept is a growing concept of how to store information in sets of ones and zeros and stuff like that. So if scientists could understand how, scientists understood how the information was stored, they would be able to change it or understand how it made us all different from one another, and how that information could even be transferred between different generations, and why it wasn't always transferred perfectly. So Rosalind and Franklin and Maurice Wilkins in 1951 were working in a lab. Maurice Wilkins was working begrudgingly with Rosalind Franklin, but who was doing most of this work, but Maurice Wilkins does have a role to play in the story. So one thing that Rosalind Franklin did was she was capturing DNA images by precipitating out crystalline DNA is what I'm assuming. It could possibly be non-crystalline, but I'd assumed it was crystalline DNA. Now I'm sort of doubting myself. But this is a very challenging prospect because polymers don't like to crystallize, and so to gather this DNA was difficult. And one of the things that she was doing was she was collecting images using X-ray technology, which was pretty new at the time, only been around for maybe a couple decades, and at least in an effective way. And so she had to learn how to use these machines, I imagine, and then she started studying these images that she was getting. And to increase her resolution of her images, she was drying out the DNA to get a nice clear image. Rosalind Franklin took two different images. She collected actually lots and lots of images. The most famous image is called Image 51, and I've attached a version of it down here. I believe this is very similar. I'm not sure if this is exactly Image 51 that's been sort of upscaled. In either case, they are pretty similar, but you can see that this sort of blurring that she was seeing up in these top and bottom parts. This blurriness is because there's water in her sample, but it gave a much cleaner impression of this sort of X pattern, which was actually really important to determine the structure of the DNA. But she didn't know that. What she saw instead was she saw this image and she said, well, that is a bad image. That's a bad X-ray image. Look at all the cool features I see in this image where I've dried out the sample. Even this example here, we have this dry sample has lots more drops in it, or lots more little black spots. And so they thought these were important. They thought that this higher resolution image or higher clarity image was very important. And so she had this image sort of off to the side on her desk, is the story I have heard. So I'll sort of tell you the version of the story I've heard. You can go watch some other videos about some other versions of that story, and it sort of changed through time, and it sort of depends on who's telling the story. But in the version I heard, she had this image, the wet form of this image, sort of on her desk with all the other images, and she was currently working on a model for the DNA, that would be what was being proposed as a triple helix, where somehow the phosphorus sort of formed a backbone, and the nucleic acids kind of stuck out, was one concept. So the amino acids would be the nucleic acids would be sticking outside of the DNA strand, and the phosphate groups would be holding everything together in a triple helix pattern down the middle. And they were using these X-ray images to try to find that triple helix image. And I guess maybe that was coming from, I see some triple symmetry here. I don't know if that's what they were looking for, but just as I have some very basic knowledge of X-ray patterning, and for me, there's always sort of a symmetry component. So essentially, my understanding is that Franklin's funding was ending, or a different job opportunity came up, and she was essentially had a deadline for her time in this lab that she shared with Maurice Wilkins. And at the same time, Maurice Wilkins was friends with another pair of scientists from across the quad in a different building, who were known as Watson and Crick. This is their last names. And so Watson and Crick, one of the two of them, I don't remember exactly which one it is, but one of them comes over and visits Wilkins in his lab. And while he's in his lab, Wilkins says, "Oh yeah, Rosalind's done. She's out of here. Look at the stuff she collected. You know, she's working on DNA too." And Watson and Crick were competitors. They were also working on the DNA model. And so Watson and Crick said, or one of them, I think it's Watson, looked over the images that she had collected, and the one that stuck out to him with his not so much trained eye in X-ray crystallography, but something about this simple pattern that she was seeing in this version with the wet DNA stuck out to him, and he said, "Wait a second. We've been over in our lab working on a triple helix model, but that model would be impossible based on this picture." Now, we don't want to become X-rayologists or whatever, X-ray spectroscopists. So I don't want to teach you all about diffraction patterns and things like that, especially because I'm not a specialist in that area. But this very simple pattern would not be possible if there was a triple helix, is the concept. And one idea I like is that this looks a lot like what we're seeing here. It's not actually a perfect map, but you can kind of imagine that there's something here about this. I believe that the spacings all mean something. So the distance between any 2 points in a diffraction pattern are based on the distances that are found in the model you're looking at, or the chemical you're looking at. And so this model really simplified things and said, hey, in this simplistic DNA model, the DNA picture, it's impossible for there to be triple helix. And a double helix would actually explain some of this sort of symmetry that they're seeing. So symmetry here, you can divide this image in half. Well, half indicates sort of some sort of bilateral symmetry, maybe. So this is James Watson and Francis Crick. They're trying to put this model together. They mostly collected and analyzed existing pieces of data. They were collecting things from all sorts of other people, and they were using them in interesting ways. One of them, I believe, is a chemist. The other one was a biologist. And Wilkins showed Crick this unpublished X-ray picture that Rosalind had collected. And they both made the same conclusion, that this is the image of a double helix structure, not a triple helix structure. Crick and Watson end up I believe they employ Wilkins to collect the same image, or they collect it themselves. They use it to publish a paper. Watson and Crick publish a paper about this model for DNA in the magazine Nature. And in the same magazine, in the next the next article in that same magazine is the article by Wilkins and Franklin that describes, some of the other fine details of this model and confirms the model, and it appears to confirm the model of Watson and Crick. So this is a very complicated issue. Is it Watson and Crick who just happen to see the right image that gets them to, come up with this fantastic idea? Would Rosalind Franklin have eventually gotten to that same conclusion if she'd studied her data, if she'd had the freedom to study her data a little bit longer? Was it only because Watson and Crick had conversations with Wilkins that they were able to publish a double helix paper as well? And was Rosalind Franklin just never acknowledged for this paper because she was a woman? Possibly, I think it's sort of an all of the above situation. They did end up using Chargaff's rules, so an example of a piece that they took from others to kind of incorporate and combine into their model, as all good science does. Science builds on other science. They used the idea of the thymine and adenine having equal amounts, and they said, well, what if the equal amounts was because the adenine and thymine were across from each other in this double helix system? And what if the guanine and cytosine was always across from each other? If they were always across from one another, then they would have equal amounts. And then they started to introduce this new concept that chemistry had just discovered called the hydrogen bond. And they said, well, what if these little hydrogens that were here were able to hold these chemicals together? And look at this. These two chemicals could make 2 bonds, but these two would be able to make 3 bonds. And that might be one of the reasons that the DNA model holds itself or organizes itself in this way, that adenine is always across from thymine and guanine is always across from cytosine, because adenine can't make 3 bonds with cytosine, and guanine cannot make, and guanine and thymine can't make 3 bonds either. There's no hydrogen up here if you tried to put a thymine across from a guanine. So this is the only way that you get the maximum number of bonds across from each other. This is the concept that becomes known as complementary base pairing. So there are 2 types of nitrogenous bases, the purines, adenine and guanine, and the pyrimidines, thymine and cytosine. And purines always pair with pyrimidines, held together by hydrogen bonds, but the adenine and thymine connect because they can form 2 bonds, and the cytosine and guanine connect because they can form 3 bonds. And this is not really used to hold, this doesn't actually hold the DNA molecule together. That's a misconception that was just recently discovered. The hydrogen bonds actually don't have enough force to hold the thing together, but they are important to organizing the DNA strand. The DNA strand must organize in this way. There's another force that holds the whole thing together. Okay, so Wilkins shares Franklin's work without her permission, which inspires the key correction to Watson and Crick's model so that they can also produce a double helix model. Watson and Crick publish a paper, and Franklin and Wilkins also publish a paper in the same issue of Nature, and the Nobel Prize is eventually awarded for their work, but Rosalind Franklin had already left the lab, and she had already had quite an illustrious career studying viruses, but then died of cancer. I believe it's a bowel cancer is what she died of, and possibly due to the X-ray exposure from her years as an X-ray technician, X-ray researcher. So she does die before the Nobel Prize is awarded. With her dead, they can't award her the Nobel Prize due to the Nobel Prize rules that they can't be awarded posthumously. So would she have gotten the award? Well, that's a problem, is that we don't have that answer. We don't know if she was not awarded because the Nobel Prize Committee is free to accept the excuse that she was dead, so they couldn't award her the award. But if she'd been alive, would she have been awarded? At that point, there are very few women who have been awarded Nobel Prizes. I suppose Marie Curie would have already had her awards. So, but there's a precedent that women don't receive Nobel Prizes as easily as men in this time. And so they're even today, to be honest. And would she have gotten the award if she had been if she'd been alive? Hopefully she would have. She certainly, if Wilkins is receiving it and she's on Wilkinson's paper, I would hope so. But it is an interesting example of a woman who was sort of pushed out of a research position by the rampant sort of misogyny of science at the time, and she ended up taking a job at a much smaller university with a much smaller budget, but a place that she apparently was very happy at. And she did have a full life, although she did die early from the cancer. So an interesting study. I'm going to post some more videos that have much more information about it. And with that, we are pretty much done the history part, and we're going to start moving forward into the future and into some of the specifics about how DNA functions. Have a great day.